So welcome everyone to our webinar titled More Mussels, More Problems, an Introduction to Zebra and Quagga Mussels. So my name is Claire Ferguson and I'm the Outreach and Communications Coordinator at the New Brunswick Invasive Species Council. So I'm going to do kind of a quick intro about who we are, introduce Avery, who is our project coordinator, who will be giving the presentation today, and doing a little bit of housekeeping. So for those that are not familiar with our organization, we are the New Brunswick Invasive Species Council. So we're a nonprofit organization based out of Fredericton, New Brunswick, although we do travel around the province. We are guided by a steering committee that is made up of many different people from different representatives of NGOs, government, industry, academia, recreational stakeholders, and beyond. And we are a chapter of the National Canadian Council on Invasive Species. So our mission as an organization is to protect New Brunswick's environmental, economic, and recreational interests from the threat of invasive species, including zebra mussels. And the main three areas of our work are outreach and education, so doing webinars such as this, doing on-the-ground initiatives, so working with partners to go out and do work related to invasive species in the field, and also acting as a hub for information sharing and also collaboration. So our presenter today is Avery Wells, who I mentioned is our project coordinator. So Avery grew up on Prince Edward Island before moving to Fredericton, New Brunswick, and in 2017 attended St. Thomas University in Fredericton. So after his first year of study, Avery got a summer job at PEI National Park and fell in love with environmental science and conservation. This led him to attending the University of New Brunswick the following year, and in 2022, he graduated with a Bachelor of Science in Environment and Natural Resources. In his spare time, Avery enjoys birdwatching, reading, spending time with his cat, Newt. So just a little bit of housekeeping. So I did want to let everyone know that this presentation will be recorded and will be available early next week for you to review and also share with others as well. I did mention earlier on, but I have muted everyone's mics for the duration of the presentation, but following Avery's presentation, we'll have an open Q&A time. So I'll turn on everyone's microphones then if you have questions that you want to ask. And if you do think of any questions during the presentation that you would like to ask, feel free to also put them in the chat section, and we will get to those during the Q&A. Just a little quick show of what you should see on your screen. So you can turn your video on and off. You will be also able to turn on your microphone at a later time. And during the Q&A, feel free to use the raise hand button to ask a question or to continue putting questions in the chat. So I will stop sharing and Avery, you should be able to share now.
Perfect. So I'm just going to bring up my presentation. How does that look for everyone?
Looks good.
Looks good. Great. Well, thank you, Claire, for the introduction, and thanks for introducing Newt, who is sleeping right next to me. He might wake up and do a surprise cameo. Who knows? But yeah, as Claire mentioned, my name is Avery Wells. I work with the New Brunswick Invasive Species Council as the project coordinator, and I'd like to thank you all for joining us for today's Lunch and Learn. Today, we will be discussing the invasive species, zebra and quagga mussels. As I'm sure many of us know, an invasive species is an organism that has invaded an area where it is not traditionally found, and it typically has some very negative impacts, both economically and environmentally for that area. Today, we'll be examining the freshwater invaders, zebra and quagga mussels. These two species first were found in the Great Lakes region in the 80s, but have since spread to other areas in North America. Unfortunately, last fall, they were found in Lake Témiscouata in Quebec, which of course feeds into the Wolistook St. John River here in New Brunswick. So with these freshwater invaders on the horizon for our province, now is the perfect time to start learning about them and what we can do. So I'll be covering their history of invasion here in North America, their life cycle and biology, as well as some of the potential impacts should they establish themselves here in the province. So we'll start with a bit of a history lesson on zebra and quagga mussels. They're native to slow-moving freshwater and brackish ecosystems in parts of Eurasia, such as the Caspian and Black Sea, as well as the surrounding areas. In their native range, they form colonies along the substrate of these freshwater ecosystems, and they're predated by the round goby, another invasive freshwater species here in Canada. Throughout the 18th and 19th century, they began to spread through parts of Europe, such as Britain and Ireland, before making their way over here into North America. It's assumed that these freshwater invaders were brought over to the Great Lakes in the ballast waters of large freighters that originated in Europe. So they onboarded a lot of fresh water from an infested ecosystem, and then when they offboarded that water in the Great Lakes, those areas became infested. They were first observed in Lake St. Clair in 1988. A group of students from the University of Windsor went out for routine surveys, and they almost just happened across a zebra mussel. Unfortunately, in 1989, another similar species was found in Lake Erie, another part of the Great Lakes region. And by the 1990s, the early 1990s, they had spread to cover completely the Great Lakes region. So they took over very, very quickly. Unfortunately, the problem did not stop in the Great Lakes. Very soon, they began to spread throughout the rest of North America. And as I mentioned, they were soon found in Lake Temiscouata. So now is the perfect time to start preparing for a incoming zebra mussel invasion. Okay, so now that we know where they come from and how they got here, let's talk a little bit about how to actually identify these two species. So as the name implies, zebra mussels have these dark zigzag patterns on their shell, as you can see in the picture on your left there. Unfortunately, these are not the best way to identify a zebra mussel because they do not always show up or show up in the same way. Alternatively, quagga mussels do not have this zigzag pattern. Instead, they have these concentric dark rings on their shell. Another way to tell them apart would be their shape. So a quagga mussel tends to be a lot more rounded, whereas the zebra mussel is very triangular in shape. In fact, if you look at the picture on the left of the zebra mussel, you'll see a very flat side. They can actually stand upright without falling over due to their shape. So that's a good way of telling if it's a zebra mussel. And then, as you can see, they tend to be quite a bit smaller than quagga mussels. So that's another way you can tell them apart. So they're pretty much the size of like a fingernail or the tip of your finger. So moving on, they have very similar life cycles and very similar biology, but there are some key differences that illustrate some of their impacts. So there's a lot of information here. I'll try and focus on the key bits of information. So for example, you can see that zebra mussels are actually able to tolerate higher salinity levels than quagga mussels. So they are more likely to be found in brackish water. On the other hand, quagga mussels can be found in much deeper depths. So they can establish colonies much deeper than zebra mussels. So you can see they can kind of occupy two different environments. Moving on, another key difference between the two of them, zebra mussels typically establish their colonies on hard or rough surfaces. So think of rocks or fishing equipment, boats. On the other hand, quagga mussels can establish colonies on both hard and soft sediments. So when comparing and contrasting the two biologies, you can really see how the two species complement one another. If there's a habitat where one might have difficulties establishing a colony, the other one fits right in. It's suitable habitat for the other. So they really cover each other's bases. Moving on to their life history. So zebra and quagga mussels become sexually mature at 1 year, and they begin reproduction in the spring once water levels reach around, or water temperatures reach 12 degrees Celsius for zebra mussels. It can be a bit colder for quagga mussels. When these conditions are met, they release their eggs and sperm into the water column where fertilization occurs. After fertilization, after about 5 days, they develop into their larval stage, which is called a veliger. Now, these veligers are primarily distributed just by the movement of water. However, they are weak swimmers, so they are able to disperse themselves a little bit, which is a bit different than native mussels, and I'll get to that later on. So 1 month after they develop into their larval stage, veligers, they began to develop a shell around the veliger. And this will actually weigh them down, so they'll sink to the bottom of the freshwater ecosystem and down to the substrate. Once they're on the substrate, they do have a small foot that they can actually use to move around to find suitable habitat. But once they find that suitable habitat, they will establish themselves there for the rest of their lives. In these areas that are suitable habitat for zebra and quagga mussels, they'll typically form some incredibly dense colonies. You can really see that in the middle picture there. In fact, some of these colonies in the Great Lakes region can actually reach up to 700,000 individuals per meter squared. So they are incredibly dense colonies. In these colonies, they find nutrients by filter feeding, very similar to the native freshwater mussels we have here in New Brunswick. So they draw phytoplankton out of the water column in order to find nutrients. Phytoplankton, of course, is the base of the freshwater aquatic food web, so it's a very important resource. These mussels, zebra and quagga mussels, are very, very efficient filter feeders. They're actually able to filter up to 1 liter of water per day per individual. So you can really just imagine how much filtration is going on in a colony such as this here in the middle picture. Okay, so now that we know how to tell quagga and zebra mussels apart, and we understand a little bit more about their life cycle and biology, let's look at comparing them to some of the native freshwater mussels we have here in New Brunswick, such as the yellow lamp mussel and the brook floater, both of which are considered species at risk. So one of the key differences between the invasive mussels and the native mussels has to do with their life cycle. So as I mentioned earlier, the invasive mussels will release their eggs and sperm into the water column. Native mussels will actually draw in the sperm through filter feeding. So that's where fertilization occurs rather than in the water column. After this, they'll release their larval stage, which is called a glochidia, and it's actually quite a bit different than the veliger. So as I mentioned, veligers are able to swim and disperse themselves in the water. However, the glochidia of the native mussels are actually parasitic. So they'll attach to a fish species to be further distributed in the freshwater ecosystem. So as you can see, invasive mussels are much more apt and capable at dispersing themselves than the native mussels. Another key difference has to do with their actual body structure. So the invasive mussels have basal threads or byssus structure, as you can see in this picture on the left. So that actually allows them to latch onto surfaces to create these colonies. On the other hand, native mussel species use their foot to actually burrow into the substrate. So this actually allows the invasive mussels to form colonies over top of native mussels, essentially suffocating them out. So yeah, not very good for the native species at risk that we have here. We had already discussed briefly some of the vectors of dispersal for zebra and quagga mussels. Of course, they can disperse naturally in their veliger form, but they can disperse much larger distances with the aid of humans. So as I had mentioned, they were likely brought to the Great Lakes in the ballast water of large freighters. So they brought water in from infested lakes and rivers in Europe. And then when they brought them over to North America, they released their ballast tanks and essentially infested those waters. In fact, as you can see on this screen, zebra mussels can actually survive in the ballast tanks for up to 11 to 15 days. So there's no rush for them to cross continents. Alternatively, it doesn't need to be those large freighters with the ballast tanks. Just small, like fishing equipment and boating equipment, can also transport these invasive species within a province. So if you were to bring your boat to an infested lake, so say you went to Lake Temiscouata, and then you packed up your boat and went to Miramichi a week later, it's very likely that those mussels are still alive and capable of spreading. So as you can see there at the bottom, they can survive up to 10 days on a boat trailer, and even more so if it's in a humid environment. The same applies to like plants or fish that might have those zebra and quagga mussels on them. As you can see in the picture in the far right, those can also aid in the dispersal to uninfested lakes or non-infested lakes. Okay, so we understand a little bit about the history of zebra and quagga mussels, a little bit about their biology and life cycle. Let's move on to looking at the potential impacts should they establish themselves here in New Brunswick. And they do have more impacts than just ruining our tennis shoes, as we see in this picture. So these impacts that I'm going to be discussing can be broken up into two main sections. Environmental, so those are impacts to the actual environment or the species that live there, or they can be socioeconomic, so impacts to recreation and industry. So I will start with some of the ecological impacts. The largest of which is their ability to actually outcompete native filter feeders, thus reducing native biodiversity in the province. So as I mentioned, they are very efficient filter feeders. They can filter 1 liter of water per day per individual, and they focus on phytoplankton, which is also the primary source of nutrients for the native filter feeders, such as the brook floater in the top left or the yellow lamp mussel in the bottom right. So essentially, their efficient filter feeding and very fast reproduction can outcompete these native mussels, which, as I mentioned earlier, are actually species at risk. So they need additional protecting anyway. As I had mentioned, the phytoplankton that they consume is the base of the freshwater ecosystem, or rather the food web. So it does not just affect other filter feeders. Some fish species, such as the white perch or the brook trout, as pictured here, also rely on this phytoplankton during their juvenile stages. So if we remove the base of the food web, it affects these fish species, as well as any animals such as osprey that may prey on them. So it does reduce the nutrient pool for just about everything in the freshwater food or freshwater ecosystem. Just to illustrate this further, there was a study done in Lake Oneida by New York. It was examining the impacts of zebra mussels on walleye, which is native there in Lake Oneida, but invasive here. And they found that after the establishment of zebra mussels, the first-year walleye biomass was actually cut in half. So very clear impact on multiple trophic levels in these ecosystems. Here are some examples of other species at risk that could be impacted by the establishment of zebra and quagga mussels. So in the top right, we have the American eel. Below that, the Atlantic salmon. Moving over, we have the short-nosed sturgeon, the Lake Utopia rainbow smelt, and the Atlantic sturgeon. And these are just a few examples. Their impacts are very far-reaching. So they can also have indirect ecological impacts by acting as ecosystem engineers. This means that they actually alter the physical habitat or the physical native habitat, which can indirectly impact other species. So one way that they do this is by increasing light penetration, by filtering out a lot of the suspended solids in the water column. You can see just on the right, just how quickly they can change the suspended solids in an ecosystem. It does not take long. Now, that may sound like a good thing, but there is a difference between filtering and cleaning. So by filtering out a lot of those suspended particles and increasing light penetration, they're actually altering the native plant composition in the area, which isn't good. That can affect fish spawning or fish foraging. Additionally, by increasing light penetration, they actually increase the formation of green algae. And as green algae dies off, it creates an environment for botulism. So it might make the water more clear, but it actually has some pretty negative impacts for the environment. Also, we've been showing a lot of images of their very dense colonies that they form. So as they form those along the substrate of our freshwater ecosystems, they actually alter the native habitat, which again negatively impacts fish spawning and fish foraging. So as you can see, they've got a lot of environmental impacts, both direct and indirect. Okay, we're going to move on to the socioeconomic impacts, starting with recreation. So as I mentioned, they formed very, very dense colonies, sometimes up to 700,000 individuals per meter squared. Additionally, they can form these colonies on hard surfaces, such as boats. So you can see in the top left there, that is a colony of, I believe, quagga mussels on the bottom of a boat. This will, of course, increase drag as well as fuel consumption. So pretty annoying and costly impacts to our recreation. Also, any engines or equipment that draw water in from an ecosystem can be even more impacted. So you can see an off-board engine there that has been completely covered in zebra mussels. Of course, that will be another costly fix for your boat if that should happen to you. And then, of course, I don't know if anyone in the call has ever stepped on an oyster or a mussel, but it is not fun. So if they were to infest a freshwater beach that a lot of beachgoers like to go swim at, such as, I don't know, like Mactaquac Park, lovely beach there. But if it were to be covered in zebra and quagga mussels, it will likely result in some injuries. And we don't want that, of course. So a lot of these characteristics that impact recreation, such as their dense colonies and rapid expansion, also affect industry. So industry can be power generation stations, agriculture irrigation systems, or even water treatment facilities. So power generation stations, such as the Mactaquac Dam, require massive amounts of fresh water to operate daily. And so they draw in this water, which allows these zebra and quagga mussels to establish themselves on critical components. An example of this would be the Monroe Power Generation Station in Michigan. So it draws 1 million liters of water from Lake Erie every day, which is insane. That's a lot of water. And then in the 1980s, 1988, zebra mussels established themselves in Lake Erie. At that time, their density in their intake canals was only around 50 individuals per meter squared. However, by 1989, that had grown to 700,000 individuals per meter squared. It's almost unbelievable just how fast they can establish these colonies, which is why I included that image on the bottom right, just to show how quickly these intake pipes can become completely clogged by these mussels. Additionally, they can affect the spillway gates, as you can see in the top left, and that can be a very costly and dangerous impact. So back to the Monroe Power Generation Station. As they infested their intake canals, a lot of critical components, such as the fire suppression system, were heavily damaged, and that's something you do not want to have damaged at a generation station. Sam Smolinsky, the superintendent at Monroe, estimated that they spent $500,000 removing zebra mussels from their cooling system alone. So that was just like a one-time fix. So it can get quite expensive. In fact, Sam later said that our plant has turned into a zebra mussel nursery. Frankly, we can't fathom things getting any worse. Spoiler alert, they did get worse. In the early 1990s, their water treatment facility became completely clogged, and they actually lost drinking water in Monroe for 2 days because of this. So as you can see, they can have some very serious impacts on industry should they establish themselves in New Brunswick. However, there are mitigation options for industry should they become infested. However, they're not always the most efficient, and they can be quite costly and labor intensive. So one option of removing zebra and quagga mussels from let's say a power generation station would be physical removal. So that would be something like pressure washing, like we see in the top right. That picture was actually taken at Monroe Power Generation Station. So you can really see just what they were dealing with. However, manual removal is not quite the most effective method of getting rid of them because it requires repeat efforts every single year. because of course you're still drawing water in from those freshwater ecosystems, which means it's just continuously becoming infested with these zebra and quagga mussels. So not the most efficient way of dealing with them. You can also install like debris screens in some of your intake canals. As I had mentioned, mature zebra and quagga mussels have a foot that they can use to move around a little bit, so these screens would keep them out. However, as we know, they also have a larval stage, so when they're veligers, they can still freely swim around, which means they're still free to pass these screens and infest the power generation station. So again, not very effective, requires repeated treatments. Some of the more effective mitigation options would include pipe replacements with anti-fouling coating or the use of biocides such as chlorine. However, these have other negative impacts. So for example, using chlorine to clean out your intake canals is very effective, gets rid of zebra mussels very quickly. However, should that chlorine enter the native freshwater ecosystem, it can negatively affect some of the non-target species that live there. Pipe replacements are probably the most effective long-term solution. Replacing infested pipes with silicone-coated pipes has been shown to be 80% effective at discouraging the establishment of zebra and quagga mussels, and this coating can last up to 7 years. So that seems like a great solution. However, these pipes can get quite expensive. In fact, I read an estimate that the Great Lakes region alone spends $250 million every year just replacing pipes due to zebra and quagga mussels. So although it's effective, it can be quite expensive. And there's another image there in the bottom right that just really illustrates how easily these intake pipes can become clogged. A lot of these colonies can reach 5 to 8 centimeters thick. So yeah, it can cause a lot of issues. All right, so that brings us to the conclusion of this presentation. As we mentioned, these invasive species originated from Eurasia, but were brought over in the ballast tanks of freighters in the 1980s. They have since spread throughout North America and are now on our doorstep here in New Brunswick. Should they invade New Brunswick, they could have a wide range of ecological and socioeconomic impacts. I listed a lot. They can have detrimental effects on industry. They can really ruin our next fishing trip. And of course, we want to protect as many species species at risk as possible. So what can we do as New Brunswick residents moving forward now that we know that an invasion is imminent? Well, one of the best strategies when dealing with invasive pests is early detection and rapid response. So that essentially means the earliest we can actually find where they're invading and the earliest that we can start mitigating some of those impacts, the more effective those mitigation measures will be. So what can you do? If you have a freshwater lake that you like to visit throughout the summer, or if you live near a freshwater ecosystem, just keep an eye out for zebra and quagga mussels, now that you know how to ID them. And if you do see them, the best thing you can do is to call us up and send us a picture, let us know about your sighting, about your observation, and then we can move forward with some management options. And once I'm done here, Claire will actually put up our contact information so you can do that. But otherwise, I'd like to thank you all again for joining us today for our Lunch and Learn. And yeah, I look forward to tackling this mussel issue with all of you going forward. Thanks. And I'll stop sharing my screen. Yeah.
Just gonna change the settings so that everyone can use their microphone. There, so everyone should have access to their microphone now. It'll still be turned off, but feel free to turn it on and ask a question. So we do have one question coming from Vicky Nash. So what fish species host their Glaucidia? Are they easily sampled, detectable, discernible on gills for early detection? Is there a potential here for anglers to collect data, maybe even paired with existing monitoring, such as at different survey sites?
That's a good question. So Glaucidia are typically very small, so it's difficult to monitor for them or see if they're, attached to a native fish species. Although if we did find a way to do that, that would be a neat way of monitoring. It would kind of bring in a new crowd, like the anglers could actually aid a bit more in the citizen science initiative to actually monitor for freshwater mussels. So it's an interesting idea, though it might be difficult. Kristen or Claire, do you have any thoughts on this?
Yeah, I just want to jump in to clarify that the invasive zebra and quagga mussels don't require a host fish. So typically, they they don't have Glaucidia that attach to a host. Their veligers are free floating in the water column. Um, there has been one very recent occurrence of a zebra mussel attached to the outside of a fish, but they're not like our native species in that there are no host fish species, and technically they aren't Glaucidia as their larva. But the there is definitely potential for anglers and pairing with any form of existing monitoring. If anybody's out on the water, we're happy to help provide training on what to look for in the water, how to report. But basically, anybody who's doing anything in the water, there's definitely potential there, and definitely get in touch with us if you think there's a way to combine forces.
Perfect. So the next question we have is related to fresh and brackish water. How will, if zebra mussels make their way down the Woolastook / St. John River, how will this impact the lower St. John River? Will they be able to survive once near or into the Bay of Fundy?
That's another great question and something we have actually been looking at. So we're really trying to find their salinity tolerances just to see what sort of environments they're actually able to invade. Uh, Kristen and Claire, I'm not sure if you have the levels in front of you, but yeah, I suppose it just all comes down to what level of salinity can they tolerate? Can they tolerate the level in the Bay of Fundy?
From my understanding, it does vary between the two species. I don't have the exact level, but I believe one of them is around anything less than 6 milligrams per liter, and the other is, I want to say, it's either 3 or 13 milligrams. I'll have to look it up, but that is also a consistent exposure. So, but that is one of the factors that we are looking at to try and see if there are certain parts of the Woolastook, the Miramichi, up in Shediac, those areas that are just naturally protected from invasion because the salt content is too high.
Another question that we have is to clarify, I think you might have discussed this earlier when talking about the environmental impacts. So the green algae that we talked about earlier, was that related to cyanobacteria?
No. So the green algae I was talking about is like a true algae. Does naturally form here in New Brunswick. Cyanobacteria is actually related to blue-green algae, which, fun fact, is not actually a true algae, but just a type of bacteria. Uh, so no, they are two different ones. I don't think it really contributes to blue-green algae as much. And yeah, another fun fact, blue-green algae is also native, not actually invasive here. We're just finding that it's occurring in higher quantities as of late. But yeah, good question.
We have another question come in. So what is the survival time for veligers in standing water, for example, in the live well of a boat?
That's a good question. Off the top of my head, I know they can survive up to what was it, like 11 days in the ballast tanks. So I assume something like that. But Kristen, you might know more.
Yeah, I mean that I haven't come across anything that's specific to the live wells, but that would be my assumption as well, is that based on what studies they've done in ballast tanks. Yeah, I just saw it. It would be temperature based for sure. They do have a temperature threshold. But yeah, we should look and see if there's any actually specific research to live wells in North America.
Yeah, a lot of good questions. If anyone wants to raise their hand and ask something via mic, please do. More than welcome to.
Maybe Avery, if you just want to touch on the clean, drain, dry, the prevention.
That's right. Yeah. So as well as just keeping your eyes open for any possible zebra or quagga mussel colonies, you can also be a bit more proactive in preventing their establishment. So we have a program we like to call Clean, Drain, Dry, where we basically just encourage anglers or anyone who spends a lot of time in the water to just thoroughly clean, drain, and dry all of your equipment. As I had mentioned earlier, they can survive on boats or on boat trailers, fishing equipment for quite a long time. So yeah, as soon as you leave a lake after doing a bit of fishing, a bit of angling, just make sure to thoroughly clean all of your boat and all of your equipment before leaving that ecosystem. We just want to prevent any cross-contamination within the province. And you'll hopefully see some more signage popping up at different areas around the province, and hopefully some boat wash stations in the near future to help with this.
Have any so I do have our information up on the screen. If you do have, any other questions, or would just like some more information about the Clean Drain Dry program or the work that we are currently doing right now regarding zebra mussels, feel free to reach out to us either through email or on our website. And we also do have a number of social media pages where we are going to try and keep everyone updated this year as we kind of go into the spring, summer season, and particularly where the zebra mussels in Lake Temiscouata were just discovered in the fall. This spring is really going to be a time where we are working with partners to go out and see, the extent of their movement and if they've been found elsewhere. So definitely reach out to us if you have any questions, comments, or would like to partner on a zebra mussel related project. So I think that is it for today. Kristen did put the link to our Clean Drain Dry page on our website in the chat. So feel free to check that out.
Yeah, please check that out. And I just want to say thank you again, everyone, for joining us, and a lot of great questions. Hopefully we'll see a lot of you at our next Lunch and Learn.
Yes, there is one person typing, so we may have a question coming. Yes, Dennis, we will definitely be in touch with you about partnering this season regarding zebra mussels and other invasive species related fieldwork. So we'll definitely be in touch with that. But yes, thanks everyone for joining the webinar today. And as I mentioned, the recording will be available early next week. Thanks everyone and have a good day.
Thanks.